Ladies and gentlemen, please take your seats. Our program is about to begin. Thank you. Please take a moment to silence all cell phones and mobile devices. Video and audio recording of any kind is also prohibited. Thank you. Ladies and gentlemen, please take your seats. Our program is about to begin. Thank you. Please take a moment to silence all cell phones and mobile devices. Video and audio recording of any kind is also prohibited. Thank you.
Good morning. I hope you all have had a fantastic start to your Ocean Sciences meeting experience. Woo! I'm loving the enthusiasm. On behalf of the organizers, I'd like to thank Daniel Avila and the Berklee College of Music for composing that original musical piece you heard coming in during the introduction. My name is Kristen Buck. I'm an associate professor in chemical oceanography at the University of South Florida College of Marine Science. And I'm honored to be up here today as the Oceanography Society co-chair for the 2020 Ocean Sciences meeting. This meeting gives us all the opportunity to share our latest findings and inspire new collaborations across ocean sciences. As you know, this meeting is co-sponsored by the American Geophysical Union, the Association for the Science of Limnology and Oceanography, ASLO, and the Oceanography Society, TOS. Each of these organizations, with the engagement of us as individuals and as a scientific community, works to facilitate opportunities to bridge disciplines, connect communities, and make lasting partnerships in science. As an interagency effort, the Ocean Sciences meeting is a great example of collaboration, bringing together thought leaders of the international ocean sciences community, from students and early career researchers to senior scientists, in order to advance the knowledge of the oceans and ultimately to improve the health of our planet. I'm excited to introduce two of these thought leaders here today for the science plenaries. A biological oceanographer and inventor, Heidi Sosik is a senior scientist at the Woods Hole Oceanographic Institution, and she currently holds the Stanley W. Watson Chair for Excellence in Oceanography. In her work, Heidi has advanced researchers' abilities to study microscopic organisms that fuel ocean food chains, interact with Earth's climate, and sometimes produce harmful algal blooms that threaten both ecosystems and human health. Heidi also serves as lead investigator for the Northeast U.S. Long-Term Ecological Research Project, and she recently stepped down from her role as chief scientist of the Martha's Vineyard Coastal Observatory after 14 years. She is active in many national and international roles. She was named a Woods Hole Oceanographic Institute Ocean Life Institute Fellow in 2003. Other accolades include the Presidential Early Career Award for Scientists and Engineers, the Ahrens Award for Excellence in Teaching, Advising, and Mentoring, the NASA Agency Honor Award, the Woods Hole Oceanographic Institute Senior Leadership Prize, and in 2018, Heidi became a Fellow of the Oceanography Society, and then in 2019, she became a Sustaining Fellow of ASLO. Please join me in welcoming Heidi Sosik.
Thank you so much, Kristen. Good morning, everyone. I feel so incredibly honored to be speaking in this session, and I thank all of you for being so generous with your time and attention. It's truly overwhelming. This is really an exciting time to be a plankton ecologist in the ocean sciences. There are people all around this room giving presentations this week that touch on the many reasons why that's true. Before I dive in my talk into my personal slant on why this is such an exciting era, I want to set a bit of historical context. And this is because many of our big questions and conceptual foundations are not new at all. In fact, they've been around for a very long time. In the early 1900s, when he referred to plankton as this blood of the sea, Victor Henson was already emphasizing the central role that they play in ecological systems, and he was pressing for systematic and quantitative study of their distribution and function in those systems. But of course, the tools that these guys had for that kind of quantitative studies were very limited. Henson and we today are concerned not only with snapshots of plankton in the ocean that you can get, say, with a net, but also with how they're changing through space and time. We think about frameworks like this, where their dynamics depend on a bunch of physics that move them around in the ocean. We'll hear all about that from Erik in the next talk. And then, of course, there are these important biological terms that represent how they grow and die. And really, these are about, I would say, equally important and difficult to quantify in nature. We know, though, that for many types of plankton, including phytoplankton that I'm especially interested in and we'll talk mostly about today, these rates of biological change can be fast. The timescales can be hours to days, and all while we're interested in the cumulative effects of these dynamics at much larger scales. So the range of relevant scales is enormous. We still make great use of bottles and nets today, but these tools really can't tackle this scale challenge. So at this point, I think maybe you see where I'm going. You can anticipate. My excitement today is linked to new approaches that I think can finally crack this nut. Some of you I know will be familiar with this topic. You think about it a lot, and you might be thinking at this point, okay, there she goes again. Heidi's a little bit slow to the party, because since the 1960s, 1970s, at least for phytoplankton, we've had incredible tools for characterizing a huge range of space and timescales, things like ocean color radiometry from Earth orbiting satellites, fluorometers that we now hold in our hand and we can deploy to get both high resolution and coverage. These approaches lump all the phytoplankton together and provide measures of the total chlorophyll. This is great because that's the primary pigment they use to capture energy from the sun and ultimately to photosynthesize. The challenge is in that lumping, though, because phytoplankton are really diverse. This is an electron micrograph of one of the smallest phytoplanktons, Synechococcus. Now it's shown in scale with some example nanoplankton, a flagellate, some of the many species of dinoflagellates that are important in the ocean. Then a single-celled diatom. The dark spots in that cell are where the chlorophyll is in the chloroplast. And then we'll zoom out to a relatively small diatom chain. These can be up to millimeter scale. Do you see the original picoplankton? It's up there, not even at the pixel resolution on this screen. And there are typically thousands, even hundreds of thousands of these tiny cells for every diatom chain in a few drops of seawater. These cells, of course, are diverse not only in their size and shape, but also their genetic and biochemical potential, their physiology, the niches they occupy, and ultimately the functions that they play in the ecosystem. We love this idea of large diatoms, like chain-forming species that supporting short food chains that efficiently export production to higher trophic levels and even to the deep sea, while we think about the picoplankton as part of a much wider web of interactions, we call the microbial loop, that's thought to be largely recycling in nature. My point here is that the different types of organisms matter a lot. Think back to making that big pile of chlorophyll that you might be using to assess an ecosystem, and then deciding it doesn't really matter if the pile's made up of blades of grass or redwood trees, which sounds kind of ridiculous, right? But that's exactly what we're doing in the ocean, only at the microscopic scale. So this is where I want to introduce flow cytometry to this story. This is a technique where seawater is pumped in a thin stream, so thin that the particles are stretched out and are going single file through a focused light source, usually a laser beam. And as they go through the beam, each particle is measured, the amount of light they scatter, how much fluorescence they emit. This is a very common technique in microbial ecology. It's used in labs around the world to study especially small phytoplankton. It's ideal for picoplankton and small nanoplankton. And it doesn't work as well when we start talking about big microplankton with complex shapes. So my colleagues and I have advanced a solution for that combines flow cytometry with video imaging. In this case, just downstream of the laser beam, the particles are exposed to a short, bright flash right when they're in front of a microscope objective, and the exposed frame from a digital camera is stored. The results are these kinds of very high resolution images, about a micron resolution, of each particle. We can see enough detail here to characterize a lot about the cells, often including their taxonomic identity. Because this approach uses very precise flow control and the laser signals to trigger the imaging, we really maximize the capture of images of targets in good focus. This is an example replay of an actual seawater sample, just a few drops analyzed this way. The black region is the whole camera field, and you see each separate trigger and associated image coming along in the flow. This is essentially the raw data, so you can see the consistent image quality. I've just held the images for a split second here so that you can see them before they go by. They're really coming along separately, but the replay is done this way so you can see. Okay, so now the fun begins. Many years ago, when I started as a postdoc in Woods Hole, I began what would end up being a long-term collaboration with my wonderful colleague, Rob Olson. Rob has many talents as an ecologist and oceanographer. He is also an absolute wizard with flow cytometry. And our work together led first to this submersible flow cytometer we call flow cytobot that targets measuring the small cells, picoplankton. And then later on, we developed the sister instrument optimized for microplankton that includes the imaging I was just talking about. So I've compressed years and lots of work into one slide here. The takeaway I want you to keep in mind is that these approaches, adapted from what we could do in the lab, but now working in the ocean, allow us to rapidly count, size, measure other characteristics of cells, and it works at the level of individual cells in many cases that we can identify. And these technologies were designed specifically for extended, unattended deployments in the ocean. So back in 2003, we first deployed Flow Cytobot at the Martha's Vineyard Coastal Observatory, then a fledgling cabled site near Woods Hole. And this allowed us to document the dramatic spring bloom of Synechococcus, a picocyanobacterium that dominates the small cells in this system. This is cell concentration on the y-axis on a log scale, so they're increasing many orders of magnitude in the springtime in this system. And I must emphasize, these are automated observations, and this time series has hourly resolution. So this was super exciting at the time, but speaking of glossing over a lot, this absolutely pales now in comparison to what we have where these observations are ongoing and have produced this incredible multi-decade time series of this organism. I need to just pause here for a moment to urge all of you who are not ecologists to think for a moment about, to have an inkling of how amazing this kind of data is for us as ecologists. There's absolutely no way we could get this kind of taxonomic, temporal resolution and extent with our conventional approaches for sampling organisms. Some of you who have not heard me talk about this before are probably wondering, okay, well that's really cool, but do you really need to measure that organism with hourly resolution for more than a decade? We get it, they're going up and down every spring. In her thesis research, Kristen Hunter-Severa showed very elegantly that resolution is extremely powerful. With that detail, we can extend the flow cytometry results beyond just counting cells, and we can begin to pose questions about the regulation of the spring bloom, not just know that it exists, but why is it happening and why does it happen the way that it does? So to do this, we capitalize on the cell size measurements that are made at the level of individual cells as they go through the laser beam, and we measure their light scattering. So for each day in the time series, this is an actual day of data, we can measure the changes over the day in the distribution of cells within this population. You can see that the cells tend to be small at dawn. The whole population tends to get larger during the day as they photosynthesize, and then they get small again at the end of the day when they divide. In Kristen's work, she coupled these data to a size-structured matrix population model. She used a statistical approach to best fit the model to reproduce the distributions in the data. And then with the model, we can compute the number of cells that have to divide in order to generate the observed patterns in the community. We can apply this approach to each day in the time series, and then we add a brand new time series, which is entirely independent from the cell concentration. So that cell concentration, again, shown in the top panel there. And now this lower panel is a time series that reflects the physiological regulation of how fast the cells can grow and divide in their natural environment. And from this, we can learn that division rates in the spring follow a very systematic function of water temperature. These results are providing us with pretty direct support for the idea that these tiny cells are physiologically limited by low temperatures in the wintertime, and that spring bloom kicks off and the limitation is slowly released when they start dividing faster as the water warms. Kristen has also shown that seasonal cycle is not as stable as it at first appears. It's been shifting. In the first decade of this time series, we observed systematically earlier spring warming, which was directly reflected in earlier Synechococcus blooms. These plots show year-day crossings for isolines of first temperature and then cell concentration. And the warming and the bloom were coming about 20 days earlier over that decade. And notably now we can show not only that the bloom came earlier in warm years, but also that was tied directly to earlier increases in the rate at which they're dividing. So again, this direct physiological link to temperature. These results, of course, raise questions in our minds about the implications for this ecosystem as warming trends continue in this region. Will the growing season for Synechococcus continue to expand, and what will be the implications of that for the rest of the ecosystem? Of course, now Synechococcus are not the only picoplankton at MVCO in that system, and as part of her early PhD research, Bethany Fowler has been showing that we need to consider the other types as well. A mixture of species of small eukaryotic phytoplankton are also important, though they're much less abundant than the Synechococcus, which you see here in green, and those picoeukaryotes in purple. But it turns out that using similar approaches to Kristen Hunter-Severa, Bethany has shown that these small eukaryotes are typically dividing 2 to 3 times faster than their Synechococcus partners. And as a result, these tiny eukaryotes contribute more to primary productivity in this system than we might expect just from their concentration. Bethany's work is also suggesting that these tiny picoeukaryotes are preferred by grazers in this system because we have the high-resolution cell concentration data and also the independent division rates. She's able to quantify group-specific loss rates. We can't be sure how much of that's grazing versus other sources of mortality like viral lysis, but the rates in total are much higher for the pico-eukaryotes, suggesting that they contribute a lot to fluxes through the food web relative to their abundance. So with these approaches, we can now quantitatively assess these important characteristics of communities and the important differences among the partners in this community. These differences, we can detect them among what we might otherwise consider to be fairly similar, small plankton in this ecosystem. Okay, so there are much more than picoplankton in the waters at the Martha's Vineyard Observatory. These examples of plankton imaged in situ emphasize the huge range of taxa that we see in the larger organisms. You see their shapes, sizes, and the detail that we can capture with this kind of automated imaging. I'm just super excited to tell you that this is the tiniest slice of the data that we have. Working here, we've been deploying Imaging Flow Cytobot, the partner to the Flow Cytobot instrument, at the observatory since 2006. This record now comprises over 250,000 samples and over 1 billion images. So this is truly big data in marine ecology, and it has prompted us to invest a lot in data and information systems, and I'm really proud to say that you can access and use all of these images and the image products that are generated from our automated workflows through the IFCB dashboard. I'd be interested to see what happens if a whole bunch of you hit the server at the same time, but check it out sometime when you have a lot of time to browse. You can also do machine to machine and download as much of the data as you want without having to use the web interface. And I want to here bring attention to my wonderful colleague, Joe Futrell, who has really made this data system his personal mission, and he works with the ultimate respect for our scientific goals with these data. I wish you all partnerships with data scientists who are willing to jump up from their computer systems at a moment's notice and go out on deck of the ship in their work vests. He's an amazing guy. Okay, so given the sheer number of images, we can't possibly look at them all. So we've developed automated analysis procedures that now let us produce taxon-specific time series for extended periods with very high resolution. So here I'm highlighting just one species, the biomass-dominant diatom in this system. So it's sort of a counterpoint to Synechococcus being the dominant picoplankton. This is the dominant microplankton. There's a lot of variability here, but if you look closely, you can see that the biggest blooms occur pretty systematically in wintertime. So very different from the picoplankton whose highest concentrations were in the summer. Once again, with this detailed data, we can go beyond just documenting these patterns, and we can begin to ask, why do these diatoms bloom in the winter? And I have to say, this question really nagged us for a number of years. We sort of waved our hands about nutrients and tried to find some physical oceanographers to help us figure out if there were more nutrients, and it wasn't really leading us anywhere. And then Emily Peacock uncovered a really important piece of the puzzle. The high-resolution imaging that we're doing made it possible to detect this species-specific parasite that we previously had no idea was important in this system, but we now know causes high levels of mortality in this diatom host. These parasites are small flagellates that get inside the diatom. They feed on the protoplasm, they reproduce inside the empty host frustule, and then they burst out, moving down the chain to infect the next one, or swimming through the water till they find another host to infect. So like I said, we had no idea that this was important in the system for years. But I want to emphasize one of the incredible powers of this kind of data set is that once Emily discovered this phenomenon several years into the time series, she was able to go back to through the entire digital image archive and show that these kinds of infections are recurrent. This lower panel shows the fraction of infection in the population over all of these years. And we now believe this is a regular and very important source of mortality in this diatom host. We also find that there's a very striking pattern, again, with temperature. This plot shows infection rate observed each day against water temperature, and the points on the plot are color-coded according to host abundance on that day. So what we find remarkably here is that there's an almost complete absence of infection when the waters are colder than about 4 degrees. These are times when there's plenty of host around to support an infection, and in fact, more than at many other times of year. And we now think that those cold waters provide this diatom a sort of refuge from this big source of mortality associated with the parasites. Our analyses also show that we can explain a lot of the overall variability in bloom magnitude, shown here on the y-axis, a measure of the magnitude of each of the individual bloom events. We can explain a lot of the variability according to the prevalence of infection by the parasites shown on the x-axis. So the biggest blooms are occurring in the cold winters when there's low infection rates. Our time series is now long enough that we have some anomalous conditions that help to sort of support this level of understanding. And a notable example is the winter of 2012. This was the warmest winter on record, and water temperatures never got colder than about 4 degrees C at the observatory. Parasites were present all winter, and the diatom bloom essentially failed. So again, we have new questions about, as climate change continues to impact this region, about what the implications of this will be. We may expect that these coldest winter water periods may shorten, they may disappear altogether, and we now have to wonder if this species that is currently dominating the diatom biomass may not be so in the future. There will be some community shifts. As a final point on this, I want to mention an important implication for ecosystem structure and function. Remember, again, this conventional paradigm that diatoms are part of these short food chains with efficient export production. But we have to now contrast this view with the role of these nano-sized parasites that I've been talking about. They're likely part of the microbial loop. So we have to consider that there's an important pathway shunting diatom production right into the heart of the microbial loop. Okay, on the topic of the microbial loop, a central component of that are protozoans. And I just want to briefly mention that in her thesis work, Emily Brownlee showed how well the IFCB imaging approach works to characterize these really important single-celled grazers. They tend to be quite fragile and difficult to study with other approaches. And at MVCO, we can produce species-specific time series for these type of micrograzers, just like we can for the phytoplankton. We typically think of these organisms as consuming picophytoplankton, maybe small nanoplankton if their mouths are big enough. But the high-resolution images at MVCO also emphasize that many types of small grazers can attack much larger prey, things like diatoms. They use things like feeding tubes and external sacs that they externally digest large prey in, or they can even stretch their soft bodies around large prey to digest them. So again, like those deadly nano-sized parasites I mentioned, this adds even more questions to the common view that diatoms are principally consumed by relatively large zooplankton like crustaceans. Now, even the basic idea that big things eat littler things may not be such a good bet. We are certainly not the first to show these kinds of phenomena, but this imaging approach with time series provides a unique way to learn more about these processes and how important they may be in natural communities. Harmful algal blooms. Considering this year's theme for our conference, I want to take just a few minutes for a sidebar. I've been talking a lot about the important functional roles of different kinds of plankton. We know that sometimes the balance shifts to bloom species that can have harmful effects on ecosystems and even on humans through production of toxins that either that can enter the food chain or have other health impacts. I want to put out a suggestion of IFCB as a small contribution to science for a resilient planet. We developed this technology really for basic research, but it has also already proven to have direct applications in HAB detection and response. The first demonstrations of this were led by Lisa Campbell's group working in Texas waters. In 2008, the IFCB time series data made it possible for them to provide an alert about a completely unexpected toxic bloom, the first of this dinoflagellate species to cause a toxic bloom in this region. So the resource managers weren't looking for it. This event did ultimately lead to contaminated shellfish that could have made people very sick, but the early warning prevented that. In this area, IFCB has also been used to show things like when small scale but very intense patches move into the Port Aransas estuary from offshore origins. These patches are so intense that are small scale that they're easily missed by intermittent manual sampling. So the continuous monitoring that IFCB can do is critical. This happens to be the species that causes the famous toxic blooms along the Florida coast, and it's increasingly a problem in Texas as well. Other groups are now extending these kinds of HAB applications around the world. Just a couple more examples around the US. Mike Brosnahan's group has been studying the ways that life cycle stages and parasites, again, impact toxic bloom dynamics of dinoflagellates that are important in New England waters. And the work of Alexis Fisher from Rafe Kudella's group highlights toxic diatoms that are commonly a problem in waters off the west coast of the US. I should note that in each of these cases, IFCB can provide information to detect HAB events, but it's also being used to better understand why and when they occur. So improving our understanding of them and perhaps better mitigation strategies will emerge. This kind of spread in the use of IFCB emphasizes in important ways that the MVCO time series is no longer unique. We've worked really hard to promote that. IFCB is now a licensed, commercially available product with a growing user group around the world. And we continue to invest in hardware improvements as well as software improvements, including pushing the envelope for automated classification so that most recently we have deep machine learning models that scale well to over 100 classes, 150 now, with excellent class-specific accuracy. We're openly sharing our analysis approaches and code bases, and I hope this will continue to stimulate more uses and new ideas. So I know that there are some of you in the audience who share my passion for time series in ecology. I'm jealous of those of you with longer time series. Some of you have heard me talk before about my 3 biological children, and then almost in the same breath, these time series. Not really, but you get my point. So they're kind of digital offspring, right? They need to be cared for and listened to, and then when you least expect it, they totally amaze you. Even the saying, it takes a village to raise a child, applies here too. There's Rob, of course, Alexi, Taylor, and Emily. I just can't imagine more dedicated technical experts. And then a growing parade of people who've really been critical to making this happen, making these data sing. And I would be remiss to not mention the array of funding sources brought together, whether they knew it or not. Their contributions have made something bigger than one project. So, you know, time series rock, right? And some of you are like nodding with me, and then I can just like feel the vibe. Some of you are like wanting to jump out of your seat and shout, hey, you know, it's a really big ocean out there. And half an hour ago, you were going on about all these spatial scales, and how can you just sit there in one spot in the ocean and pretend you're learning important stuff. And I feel it. And I'm so thrilled to tell you that a new NSF-supported long-term ecological research project means that we are not only able to continue the MVCO time series, we're also going to be continuing our show on the road strategy, where we're setting up IFCB to sample, and also conventional flow cytometry, but to automatically sample from underway seawater systems on research vessels. We are focusing on a section cutting from MVCO across the shelf to the edge of the continental shelf, where we are able to leverage the WHOI Pioneer Array, which is located on the edge of the shelf in this region. And we are undertaking these surveys seasonally, so we have winter, spring, summer, fall in our first year. But I just want to come back to focusing your attention on winter now. And these maps are showing the wintertime distribution of Guinardia delicatula, first along the transect, and then also we're sampling an even larger broad scale within this project by partnering with the National Marine Fisheries Service on their surveys that support ecosystem-based management in the region from North Carolina to Maine. So this is now spatial maps of that same diatom species that I've been talking about being so important in the MVCO time series. And we can now begin to see that, yes, that same species occurs at high levels in many areas throughout the region in winter. And going forward, I'm really excited that we'll be able to work to evaluate how well we can build on our knowledge from MVCO to scale up to understanding the dynamics of this and other species throughout the region. So it's really great when we can extend our existing understanding like that, but it's actually even more fun. We're already finding some big surprises. I mentioned a while ago that the MVCO time series is greater than 1 billion images. You may have noticed every time I've showed this time series that there are red points superimposed on the high-resolution blue time series from the automated analysis. Those points are validation of, from the labor of love led by Emily Peacock in my lab. Emily has inspected 4.7 million images. Every 2 weeks, she randomly chooses an hour of data, and she annotates every image in that data. We're now labeling about 50 types of diatoms that exist in the MVCO time series. And this important one, Guinardia delicatula, makes up 81,174 of the current annotations. I highlighted that species in red there in the center, because now I want to draw your attention to the one right below it in this mosaic. This is a genus Hemialis. And for those of you who don't love phytoplankton, it looks about like Guinardia, right? It's a long diatom chain about the same size. How many of those do you think we saw in those 4.7 million images that Emily has looked at? Just give me a number. What do you think? 42. Oh my God, give that guy a prize. The answer is 37. 37, okay, it's like a joke, okay? Not a diatom we were paying any attention to at all. Not important in this system, okay? That's what we thought. Then along comes the August 2019, just a few months ago, summer transect for the LTR project. And boom, these are just some of the images. I didn't show them all here, you wouldn't be able to see them. In one 3.9 milliliter, so this much water, chock-a-block with Hemialis, the jackpot. This is the concentration plot for Hemialis along the transect. All those bright yellows, they were everywhere except the inner shelf and the upper slope. It was just chock-a-block with Hemialis. This bloom was widespread and really rocking, lots of healthy cells, evidence of cell division. This species was really dominating in the waters. Now, when we look more closely at the images, some of you who know Hemialis will be expecting this. It's hard to see in the healthy cells, but when they're a little bit damaged, we can see that the host diatom cells have these partners. There are nitrogen-fixing cyanobacterial symbionts that we can see in the images. We have to add a new class to our training set, rebuild the classifier because there's something new. And we now are presuming that this partnership has provided this bloom with a completely different nutrient source than the other diatoms that we typically see in this region. This is a trick Guinardia delicatula can't play. So these highly stratified, very warm late summer surface waters may be just the niche for this kind of association. This is a quick TS plot, temperature salinity plot, across all the Transect cruises, showing that this bloom we encountered occurred in the warmest shelf waters we've sampled to date. So now we have new questions. How often does this occur? Is it always in summer when the water's super warm? What impacts does this have on the rest of the ecosystem? I want to make a quick shout out here to Samantha Sita. Many early career scientists participate in this LTTR project so far, and Sam made a Herculean effort. She volunteered within less than 24 hours notice to join us on this cruise because we had a late breaking schedule change and some people couldn't go. And it was fantastic to have her along, a training experience for the early career scientists. But she was the one who knew what these symbionts looked like and found them in the images and led us to be thinking about this in the middle of the cruise. So kind of great partnership there, and we love having those students involved. So we've really only scratched the surface of the stories that the MVCO time series has to tell us. And I've just highlighted what I think are a couple of exciting results from a few of the cruises. Our sampling of the region already looks like this. Each point here is an IFCB sample containing hundreds, even thousands of images, and the different colored tracks are different cruises through the region multiple times per year since we started this partnership with Fisheries Service in 2013. And the beauty here is that this is just the beginning. This kind of sampling is sustainable. And I really hope you'll stay tuned to see what the next decade uncovers as we dig into this huge data set. Over the coming decades, I'm sure there will be more exciting advances in observing tools that we'll be watching for as well. We're already hearing about some of them at this meeting. In the meantime, today is really exciting because we now have the tools that we need to meet the spatial and temporal scale challenge I laid out at the beginning, while also providing an aspect of taxonomic resolution that was really previously only possible with manual analysis of discrete samples. Taken together, this is already having societal impacts, and it's accelerating the pace of discovery, leading us to new understanding, new models, fueling new hypotheses about how ecosystems function, what their links are to climate, and so on. I wonder what Victor Henson would, how he would react. He might have been kind of a curmudgeon, but I hope he would have been amazed and excited at these new insights into his blood of the sea. Thank you.
Thanks, Heidi. All right, you guys, we can take some questions. So the way we've worked this out, there are 3 microphones, creatively numbered 1, 2, and 3 here in the center aisle. If you would like to ask a question, please make your way to one of those microphones. And I can't see faces or badges, so I'll call on you by number, and we'll go from there. All right, we've got a question at number 1.
You are clearly a god in this field.
Maybe I'm a goddess.
Yeah, well, that's true. I didn't see your pronoun button. And you have shown a tremendous ability to perceive and resolve issues on the scales that you're talking about. And I myself have Synechococcus and diatoms, and we look at them, and we have one issue that you showed a picture of but did not address, but I believe that you are the person to lay to rest the problem. And that is, our peaks are at 30 to 50 meters. What do we do with deep algal blooms?
What do we do with them? You mean besides study them? Oh, surely they're not, right? All these amazing phenomena in nature have to have some, it's all interconnected, right? And I'm sure they're important ecologically. I didn't have a chance with the time available to show that we're also doing depth resolved sampling on those LTR transect cruises, and we're able to look through the seasons and through multiple years at the differences between community structure and biomass of different species in the surface waters and in the deeper waters, and that will definitely be problems we'll be exploring more. And I'd love to talk to you about your thoughts about what the right way to go about that is. I'll also say, I know there are more questions, but I don't feel like a god or a goddess. I feel like so blessed to be surrounded by smart, motivated people who just sort of make it all happen and make it fun. Sorry, you can.
Heidi, great talk as always, and it's, as you say, it's a huge work of all the community, and what I really enjoyed is bringing together this technology that you talked about in the bot, and also the analysis system, deep machine learning and all of that. So it really opens up a new window in this complicated marine ecosystem. But I wanted to challenge you projecting the thoughts of the next 10 years. I mean, spatial resolution is just enormous that you now have by having that on a ship. Maybe it'll be on robots soon. You're going to see more of that. But I want to push you a little bit ahead and say, I'm also hearing from other colleagues, none of this is my discipline, I'm just a poor physical oceanographer, but there is these studies about eDNA and looking at the ecosystem from a different lens. And could you maybe say just a few words how I should understand your approach in the context of that approach?
So again, I'll come back to the being blessed with being surrounded by smart students and early career scientists who are approaching just those strategies in the context of the kind of, say, image and cytometry-based sampling that we've been pushing for so long. So I've worked with students who've already begun to explore the let's say, the space of what you can learn, what more you can learn, what different things you can learn by bringing together cytometry, imaging, sequencing, studies in the laboratory. So I didn't have time to get into it, but one example is Synechococcus. I talked about it as if they're all the same. It turns out there are, as many people in this room know, there are many different genetic types of Synechococcus, and we know at the MVCO time series from studies that some of my Kristen Hunter-Severa has started that there's seasonally, there are very distinctive shifts in the community structure within the Synechococcus. We can't see that with conventional flow cytometry, but when we bring together sampling of the at the genetic level with the high-resolution flow cytometry, I hope we can take the next step in terms of understanding what parts of the dynamics are regulated by different, say physiologies. And Synechococcus, that's just one example. Within the LTR project, we have a whole component that's led by Tatiana Reinierson that's focused on genetic characterization of across the plankton. So we will be bringing these things together. It's going to be a great next decade.
Please. Hi, Heidi. Thank you for that. It's wonderful. So a little bit like Martin, looking forward, what are some of the technical challenges? You made it look like everything works all the time. Tell us some stories about what didn't work, and then look forward 10 years, and what do we have to overcome?
Okay, what didn't work? I mean, gee, so I'm sure some of you saw a few gaps in my time series, right? So like just one example, in the early days, we were using syringes that we were terrified to deploy in our instruments in the wintertime because the cold waters caused the seals to shrink and the syringes would start leaking inside the instruments, and that's not good, right? So there was a challenge in the early days of developing these instruments to work with the manufacturer of the syringes, come up with cold-tolerant syringes. We have to pay more for them, but we also learned over the years which our deployments don't end due to fouling because our instruments keep themselves clean. They also run their own internal standards, so they monitor their own performance. But they do end because eventually some mechanical part which just wears out after thousands of hours or going up and down or turning. We started installing duplicate pumps, so one of the pumps that would would routinely fail. We now have 2, so we can switch to the second pump during the same deployment and not have to end it. The challenges going forward, there's so many. There's so many strengths of this cytometric and imaging approaches that I talked about, but they also have limitations. It takes us many minutes to analyze a sample and get statistical information about the community structure. The current depth rating of these flow cytometers is only 40 or 50 meters, and the point about deep populations, if we really want to get more in situ observations, we need to push the envelope on getting the cytometers deeper. We want to bring them together with other kinds of imaging strategies that let us look at both smaller and larger organisms in the community. So an example, in the LTR project, we're now on the Transect cruises using shadowgraph imaging to look at the zooplankton community at similar scales that we can characterize the phytoplankton community. We always want the sensors to be smaller, lower power, easier to deploy. There's a lot of work to be done. Bring it on, engineers. Sorry, long-winded.
Thank you for that talk. I'm not a biologist either, a bit like Martin. So you showed the equation at the beginning, and you talked about the growth and death rates of the various classes. Have you got enough information this moment out of that data set to actually estimate those growth and death rates as a function of all the other things from that data set to go and do that sort of predictive calculations?
So if I understand your question, I think you're referring to the ways that the growth and death rates depend on other factors that drive them, like temperature, nutrients, light, something like that?
Well, even can we estimate the growth and death rates? Forget from the collection that you have, right? Form that equation to do some prediction.
Yeah, so I showed some examples for specific groups of actual estimates that we're producing from the cytometry. We've also, in collaborations, other estimated division rates for the larger organisms based on the image data. So some of those vital rates we're getting directly from this sort of deeper analysis of the cytometry, not just cell counting. So I'm not sure if that's answering your question. We can try one more time, and then maybe we can...
In fact, you are.
Okay, good.
So can we go forwards then and just plug them into that equation and project?
Yeah. So the challenge of, I think, is, I presented results at the level of individual taxa, because I think ecologically that's where we have to be focused in order to understand the underlying dynamics. When we start aggregating everything and measuring, sort of emergent growth rates of the whole population, it becomes very difficult to understand because there's so many different facets that are contributing to those net results. So the challenge going forward in terms of, I think what you're suggesting are models that we can use to hindcast or forecast, predict the responses to changing conditions, for instance, is going, as many people in this room are working on this challenge of how much detail do those models need to represent in terms of, say, functional types or taxa. Is it sufficient to have, small cells and large cells? Depending on your question, maybe not, because, some of the large cells have nitrogen-fixing buddies and some of them don't. Some of the small cells are growing 3 times faster than the others. So, and I don't pretend to have the answers about how we sort of thread that needle in terms of bringing together the level of biological detail I talked about today and, global models of biogeochemical cycles where we're representing plankton functional types. But that's another big area that the next decade is going to continue to contribute to.
Okay, we'll do one last question.
Thanks so much, Heidi, for a great talk. I think I might ask you to repeat some of what you just said, but given the stories that you told and examples, for example, of the role that an understanding of the organism, for example, an understanding of these nitrogen fixers and what they look like, how that shaped your findings. I'm wondering what your view is on how natural history and an understanding of the intricacies of these organisms will shape what we can do with IFCB moving forward, things like the machine learning algorithms and so on.
Okay, so how an understanding of natural history will shape what we do with machine learning algorithms? Like, is that kind of where you're going?
I guess more generally, we have all these detailed and wonderful images, and how does that intersect with our knowledge about these organisms and what we should be looking for and recognizing in those data sets?
Yeah, I mean, I'm not sure I have an answer to your question. I can just say that, I mean, I think that's really one of the most fertile and exciting grounds here. I kind of like to think about these approaches that we're taking. In some ways, it's like natural history on steroids, right? And we're exploring things about the natural history of organisms in new ways that are going to uncover things that I think that the traditional, just looking occasionally through a microscope kind of natural history makes very challenging. Yeah. Again, how we bring that forward to larger understanding of the ways that level of detail can be, for lack of a better word, I'll say parameterized to reflect an understanding of larger function of the system, the sort of emergent characteristics like productivity or, export to higher trophic levels. That's one of the grand challenges I think we're going to still continue to grapple with for some years. We're fortunate to have a lot of smart students that thinking about things like that, and I'm looking forward to hearing from them.
Yeah, indeed. Well, please join me in thanking Heidi.
Don't want to forget my old lady glasses here.
All right, so now joining us is Erik van Sebille. Erik has worked in the US, Australia, and the UK before returning to the Netherlands. Now a physical oceanographer at Utrecht University, Erik studies the pathways and timescales on which ocean currents transport water, nutrients, planktonic organisms, and even pollution such as plastic. In 2016, he received a European Research Council starting grant of 1.5 million euros. It's a nice way to start it off. To investigate how plastic moves in the ocean, and he leads a team creating a 3D map of marine plastic pollution. Erik has a passion for science communication. He's done more than 300 interviews about plastic pollution and advised UK Parliament and the European Commission on the problem's scale. In 2019, Erik was awarded the Utrecht University Publi Prize in recognition of his engagement of the public in his research. Erik was admitted to the Young Academy of the Royal Netherlands Academy of Arts and Sciences in 2018, joining a fellowship of top-level early career scientists who are visionaries in science and science policy. He was recently named a 2019 James B. Macelwane Medalist by AGU, an award given to early career scientists based on their depth and breadth of research, impact and creativity, as well as service, outreach, and diversity. And he was also awarded the Outstanding Young Scientist Award from the European Geosciences Union's Ocean Division. So he's received these top honors from both the European and American geophysical unions. So please join me in welcoming Erik van Sebille.
Yes. Thank you. Okay, I just need to turn this on. Well, thank you, Kristen, for that fantastic words. Thank you all for being here. You are with so many. This is awesome. Wow. Okay, this is the ocean. This is the Southern Ocean as you see it, and this is a spaghetti. It's a spaghetti of virtual particles that have been moving around with the ocean flow and have been going around in the Antarctic circumpolar current as you see there. But on top of that, on top of the general patterns that you see, they've also been moving in much more fine-scale patterns, almost turbulence. Now, it's these kind of patterns that as an oceanographer I'm interested in. I'm interested to figure out why the ocean moves stuff around, how the ocean moves stuff around, and what that means to the global scale transport of heat, nutrients, plankton, and plastic. Now, this is going to be all about ocean models and about virtual oceans. But of course, as Heidi so eloquently said, we really also need the observations. So I think that here as a modeler, I stand on the shoulder of all these observation lists that have ground truths everything I said. And only because of that can we now are we in the age where we can actually do things with models that we trust enough to make predictions of how our ocean is going to be in the future. But let's go back. Let's go back 20, 30 years ago. It was the time of the first global large-scale ocean circulation climate models. And there was a team in Europe with Christopher Deus, Sibren Drijfhout, Bruno Blanke, Sabrina Speich, Bob Mars, many others. And they started to inquisite those global 3D ocean models to figure out how water parcels move around them. Because they wanted to trace out the conveyor belt circulation. So this is a fantastic animation that was made in 1995. It's really gone a long time. I mean, I was still in high school at this time when these people started to probe their ocean models to figure out how the ocean actually moves stuff around. And this is one single particle that they put there, the white dot, just off the coast of Chile. And as it moves around, it first crosses the entire Pacific Ocean, going west, bit of retroflexion into the tropics, and then very quickly already goes through the Indonesian through-flow, into the Indian Ocean, circulating around the southern Indian Ocean a while, then into the tropical Indian Ocean, northern Indian Ocean, back into the southern Indian Ocean again, and then at some point getting into the Southern Ocean, into the Antarctic circumpolar current. And there does this loop. And it loops around and around and around. The color scale is the depth of this particle. It doesn't have a color bar, so I still don't know really what depth it is. But there's some information in that too. It very quickly goes through the South Atlantic and then into the North Atlantic, where it does one final loop of the subtropical gyre in the North Atlantic before finally reaching the UK coastline there. Now this is a 3-degree climate model. This is really, really old school. This is as diffusive as tarmac in some ways. But still, the Lagrangian view of this is elucidating. It is telling us something about how the ocean moves all this water around. Right? So even back in the days, people really had a use for understanding ocean circulation, not only in the what we call the Eulerian framework, where essentially you look at one location and you see the flow pass by you, a bit like what was done in Martha's Vineyard Observatory. But here we're going with the flow. We call that Lagrangian. And I will come back throughout the rest of this talk. Now, fast forward 25 years, and the type of simulations that we have now, they're 1/12 of a degree. So they're almost 50 times higher resolution than that previous one. And this is a simulation that we did where we just put virtual particles just at the surface of the ocean. It's not in 3D. Here, on purpose, we put them, we fixed them at the surface. We started them everywhere, uniformly in the ocean. And then this goes something like this. See that over time, the tropics very quickly clear out. The virtual particles move away from the tropics. They also move away from the Southern Ocean. And they move into these areas in the subtropical gyres. Those are called they are the subtropical gyres. And you may recognize that those are also the areas that are now sometimes called the garbage patches, the accumulation zones of plastic. And that's exactly why we did this experiment, because we wanted to know how plastic accumulates at the surface of the ocean, and why the amount of plastic on the surface of the ocean is not uniform over space. Now you see that there's these garbage patches that form at 30 north and 30 south, roughly. And it's not only in a model. These garbage patches have actually been observed in real world too. So this is a map from a paper by Carole van der Loo and colleagues who went out with a ship. And they towed a plankton net behind the ship, like in the photo here on the right. And this plankton net, after they trawled it for a mile or so, they filled out the contents, they removed all of the biological material, and they ended up with plastic. And then some poor undergraduate student had to count all these pieces of plastic. And that is this map that you see here. Something like 4,000 or so of those samples is in this map. You see the west coast of the US and Mexico there. And blue means that there's almost no plastic floating on the surface of the ocean. So fortunately, most of the tropical Pacific Ocean is still rather devoid of plastic. And so is the high latitudes. But you also very clearly see there this bullseye area, going up to red and even purple, more than 500,000 pieces of plastic per square kilometer. Now this is the garbage patch. This is the accumulation area between Hawaii and California, just off the coast of where we are right now, that people sometimes have called the islands of plastic. You don't want to know how often I've been asked whether I've ever stood on the island of plastic. As if you can go there and plant a flag, right? It's nothing like that, though. And I can't blame people that they think that, because very many of us would have mental images of these garbage patches to look something like this. But this is not a garbage patch. This is called a ghost net. It's an abandoned fishing net. It's extremely detrimental. It is very, very bad for marine life because it constricts fish. But this is, even though it floats around in the ocean, this is not consumer plastic. This is not the reason why we take recyclable cups to the coffee places these days. This is not the reason why we stop using straws. There are no cups and straws in this figure. So if you want to have a better feeling for what the accumulation zone, the garbage patch, what it actually looks like in the real ocean, then I think that this is a much better photo. This comes from a paper by Charles Moore, who went out into the Pacific accumulation zone between Hawaii and California. And indeed, he did this thing where he trawled a manta trawl for 1 nautical mile through the ocean, emptied it, removed all biological material, and ended up with this, a petri dish full of tiny pieces of plastic. Some of these pieces you can still recognize. There's a bottle cap ring there, quite clearly. Below that in pink is probably a piece of straw. But most of the pieces of plastic are completely unrecognizable. They're fragmented, they're small. We can't really know what they are. We know the material, but not the item that it used to be. We have no idea of its origin. We didn't know where it comes from. We don't even really know its age of all these pieces of plastic. The only thing that we can really do with these is count them. And indeed, if you count the number of pieces of plastic in this petri dish, and you know that the trawl has been trawling through a mile of ocean, and it's a width of roughly 60 centimeters or so, then you can convert this to number of pieces per square kilometer. And if you do that, then in this case, you indeed get to almost 1 million pieces of plastic per square kilometer. And it sounds like a lot, until you realize, of course, that there's 1 million square meters in a square kilometer. So here, in the middle of the garbage patch, in the middle of the accumulation zone in the North Pacific, this island of trash, we're talking about 1 piece of plastic per square meter. I would argue that this conference center is actually more polluted. If we all go on our hands and feet here and look at the ground, we would find more plastic here probably than in the middle of the Pacific Ocean. You know, people sometimes call it a plastic soup out there. I'm really trying to get a new phrase in for it. I think we should start calling it the plastic bouillon. Because it is really like a very, very thin mist. Tiny, tiny pieces of plastic floating around in this enormous ocean. So, it's not only this trawl, it's not only the 4,000 or so that I showed on the map before. A few years ago, we teamed up with a group of international colleagues, and we came up with an inventory. We found as many of those trawl samples as we could from across the literature. And in total, we found 11,000 of those trawl data points. As always, in physical oceanography, or in oceanography in general, I guess, it's the Pacific and the Atlantic that get all the love. The rest of the ocean is fairly forgotten. But we do have some data there. And there are fortunately still people going out and measuring the other oceans that matter too. Now, once we have this data, we could combine it with models. We could combine it with flow models and did some fancy regression and machine learning, if you would. Well, I mean, we didn't call that, but essentially it is this. And with that, we came up with this. This is the map of where the floating plastic floats at the surface of the ocean right now. This is probably your best estimate of where all this small plastic, and with small I mean anything that fits in a manta trawl, where that is. You see in blue, very low concentrations, fortunately still around Antarctica. Also fairly low concentrations around the equator, where there's divergence, so the flow is away from it. And high concentrations indeed in each of the 5 subtropical gyres, and especially in the North Pacific. But you also see very high concentrations around Southeast Asia, closer to the sources of a lot of this plastic. And, shockingly, highest concentrations in the Mediterranean. The number of pieces of plastic in the entire Mediterranean in this analysis is just as large as the number of pieces of plastic in the entire North Pacific. There's an enormous large number of plastic there. Now, when we have a map like this, then it's fairly easy to just sum this all up, to integrate it and to ask ourselves, how much plastic is floating on the surface of the ocean right now? When we did that, we got to this number. The total number is something like 15 to 51 trillion particles, weighing 93 to 236,000 metric tons. And that's a shocking number, especially the last one, the weight. It is shocking because it is so extremely low. Well, why is it low? Well, let's do a bit of a budget analysis. From this analysis and also from other published studies, we know that there's not really much more than a few 100,000 metric tons of plastic floating at the surface of the ocean right now. That's the plastic that we've mapped, that we know roughly where it is. But there's other estimates that say that there's at least 5 million metric tons entering the ocean in a single year in 2010 alone. So in that year, 20 times more plastic entered the ocean than is floating at the surface of the ocean right now. And that's going on year after year after year after year. I would say that really only 1% or so of all the plastic that is in the ocean we've actually mapped. We know where it is at this moment. 99% of our plastic is missing. You know, astronomers, they talk about dark energy. In the ocean, we have dark plastic. Except for it's not really dark, and it's not like the astronomers who really have no idea where it is. Our problem is actually that every time we go and look for plastic, we find it. Colleagues of mine who go to the ocean floor and sample the sediment and look for plastic typically find plastic in the sediment. We now also, well, we've recently sampled the South Atlantic gyre at 3,000 meter depth. We find pieces of plastic. We all here in this room, if we've ever gone to the beach, and looked very carefully, we will have seen plastic. There's now more and more evidence there's actually plastic that is so small that it can't be captured by those manta trolls. We start calling that nanoplastic these days. And then there's the thing that should really worry us all. That's the plastic that is in biota. Fortunately, we think it's not very much yet, but this is, of course, a reason that we care about the plastic. If the ocean was devoid of life, if it was just a bathtub, then would we all be making such a fuss about all this plastic in the ocean? So we know, and we've documented, that there is plastic in each of those reservoirs. The key point is that we have absolutely no idea how much is where. There really is no one in the world that can tell you with any good faith whether there's more plastic on the ocean floor or more plastic in the coastlines. We really have no idea. These are big question marks. And if we don't know where all this plastic is, how can we ever start thinking about doing something against it, about cleaning it up? How can we clean up things where we have 99% of it is missing? And more importantly, if we don't know where the plastic is, how can we ever measure its harm? How can we ever measure where the plastic does harm to marine life? Again, marine life is not uniform in the ocean. Plastic is not uniform in the ocean. So really what we should want to know is where the overlap is between the plastic and the marine life. And in order to do that, we need censuses of marine life, of where organisms live, but we also need maps of the three-dimensional distribution of all this plastic. And that's exactly what I'm doing, or what we are doing in this project. And this, coincidentally, was figure number 1 in my grant proposal. So I promised the European Union that I would put numbers on those question marks. Yeah, I get shocked too every time I see it. But we're making good headway. So what we're doing is we're developing these virtual simulations of plastic moving around in the ocean. And what we do is just like the animations that I started with of particles moving around on the ocean, and now we're doing it in 3D. But it's not just passive particles. The particles, they beach, they fragment, they sink, they ingest, they get biofouls. All of that is part of this three-dimensional simulation of virtually polluting the ocean, essentially. And then we're combining that with as many observations, as many measurements of plastic concentrations in all of those, of the different reservoirs, to essentially to train the model, to make the model better and to make it as close to the observations as possible. And once we've done that, and hopefully we have some kind of idea of where all this plastic is. So the plastic in the ocean is an atrocity. I think we should be ashamed as a society that we've let it gone so far that our ocean is now full of plastic. But as an oceanographer, I think there's also a silver lining to that plastic. Because the plastic is also an opportunity. It's an opportunity to learn more about how the ocean works. It's an opportunity to hone our knowledge and to improve our knowledge of the interaction of flows and the movement of stuff around the ocean. Plastic is a unique tracer. It has different properties from any other tracer that we know. It has different sources from any other tracer that we know. And it is with that idea that we maybe can learn something of the ocean by looking at the plastic, that we started a SCORE working group a few years ago. And just yesterday, a paper came out with a very long list of authors, many of here are in the room here, where we make exactly that point. That plastic is unique and a fantastic tracer to study the interaction of the very, very small scale with the very, very large scale in the ocean. On the one hand, it is the gyre circulation, the gigantic 1,000-kilometer scale circulation. But then on top of that is the very, very small scale from individual waves and the mixing in Langmuir circulation and in windrows and how all that combines. And really, hopefully we will have plastic in the ocean long enough, but not much longer, then we need to solve this question, to solve the question of how scales interact in our ocean. As an example, so this is a paper that came out 2 years ago where there was a French group and they took a ship into the North Atlantic subtropical gyres. That's this map that you see in the top left corner there. And as they moved the ship, at each of those dots, they trawled again for plastic with a manta trawl, which is a standard, that's kind of the default measuring technique that we have now. But the key thing, what they did there, is that they measured and then combined it with the sea surface height from altimetry, from satellites. And what they found, so blue is low sea level height, so cyclonic eddies, low pressure areas. Red is high sea level height, so anticyclonic eddies. And the amount of plastic in the nets varied by almost an order of magnitude between nets or samples that were taken at high sea level height compared to low sea level height. So even though all of these were in the subtropical gyre, they were all in the accumulation zone, there was an order of magnitude difference between the different stations just depending on the sea level height. Now this is a NS1 study, and I was a co-author also on this. I'm still surprised that it actually got through review, right? We had one cyclone, one anticyclone, and that was it. If the reviewers are here, thank you very much for that. But it does show the heterogeneity, and it does show, and it does point us to a way in which we can hopefully learn something more about how accumulation works at the surface in cyclones versus anticyclones. And I think really we should be expanding this. And I'm sure that people in the room here, some of you are thinking about doing this now for all the cyclones and all the anticyclones in the world. So let me take a step back then. And make the point that it's this Lagrangian tracking stuff around the ocean is a very, very powerful way of understanding ocean circulation. It's not only the plastic, but it's many, many other applications. And I'm, at the moment, writing an undergraduate textbook with Bob Marsh from the University of Southampton. It's going to be my first textbook. There's a first for everything, right? About the ocean and physical oceanography, but really from this Lagrangian viewpoint, from the way of how ocean currents move stuff around, how ocean currents move marine plastic, how ocean currents move planktonic organisms, or actually really non-planktonic organisms too, how they control shelf exchanges of nutrients, how they transport heat into the tropics and other places, how they control North Atlantic water mass formation, and how the water in the North Atlantic and the convection, how that overturning circulation can also be viewed in a Lagrangian framework. How ice transport is a Lagrangian phenomena by itself. It is the movement of stuff by the currents. How river runoff is, can be viewed Lagrangian. Interocean transports. So here, for example, between the Indian Ocean and the Atlantic Ocean, and the connection between different ocean basins. And finally, really the global three-dimensional transport of the ocean. How all of this can be viewed in a Lagrangian framework. And I'm not saying that it should only be the Lagrangian framework, but I think that it is a nice augmentation to the Eulerian framework. And that really when we start combining them, that we can learn more from the ocean. Now this is one example that I wanted to highlight. It was a bit hard to pick which example I want to highlight, but I think that this one is quite nice because it shows a lot of features. This was a paper that I did out of my, my fellowship in Australia with quite many in the room here. Their names are at the bottom there. And we used a high-resolution ocean simulation of the flows around Australia and through the Indonesian passages to figure out, well, what controls how water moves from the Pacific Ocean into the Atlantic Ocean. And what we did was, in this high-resolution simulation from the Geomar Institute, we put virtual particles, like the particles that I started with, that travel the ocean. But here we've color-coded them. So the particles that started on the northern hemisphere and go through the Indonesian through-flow, we call them, we made them red. The particles that start on the southern hemisphere and go through the Indonesian through-flow are blue. And then finally, the particles that go around the south, the Tasman leakage, are green. And the key thing is we started many particles on these different sections. But we only selected those that actually ended up on the Indian Ocean. And this is the idea of conditional pathways. We can simulate hundreds of millions of particles and only select those that actually follow conditions. Say they start in the Pacific Ocean, they end in the Atlantic Ocean. And for the green ones are the ones that go around the south of Australia. Now when we started the simulation, you see indeed that they move all nicely through the Indonesian through-flow. The green ones move around the south. You see some of them are carried in eddies. You see beautiful tropical instability waves in the tropical Indian Ocean, for example. And this is just a nice animation to look at, but you can do things, for example, like look at the connectivity. The connectivity in the individual passages of the Indonesian through-flow. So this is an example that shows in SVERDRUP, so in million cubic meters per second, how water goes from each of the three straits at the top, the Karimata Strait, the Makassar Strait, and the Molucca Strait, and how then goes through the 4 straits at the bottom. And this, of course, clearly shows how the different flows are connected. We even did this in ENSO, in La Nina and El Nino conditions, all of that we compared. So this is about the connectivity between upstream and downstream flow. But these were 3D particles. And we know in the model, we know the temperature and we know the salinity of the water. So the other thing that we can do is probe what the different water masses are like upstream at 180 degrees east at the dateline. So here you see in temperature salinity space, in red, all the particles that are north of the equator and go through the ITF, all the blue are south of the equator and go to ITF, and the green is the Tasman leakage. You see that the southern equatorial is just a little bit saltier than the northern hemisphere equatorial. And then we can also do the downstream at 95 east, and you see that they've mixed. The blue and the red are not different anymore, they're completely overlapping. So this shows that all of these connections in the Indonesian through-flow, all these different straits, all this mix, it really mixes the two water masses into one undistinguishable water mass. But because we have the particle trajectories, we can still identify that. So this is from physical oceanography, and this is what I was trained to do. I am a physical oceanographer by training, but I think that the real power of Lagrangian oceanography is when it applies to other fields. When it applies, for example, to paleo-oceanography. So this is I was once invited to a workshop on paleo-oceanography, and especially then about sedimentology. So paleoceanographers looking at sediment cores and looking at the shells of foraminifera in this case. Foraminifera, there aren't so many. I mean, Heidi didn't even mention foraminifera in the ocean, but they're important because they have a shell that stays preserved in the sediment. So by looking at this shell, and then particularly by looking at the isotopic signatures of whatever this shell is made up in the sediment, paleoceanographers can say something about past climates. Past climates on scales of hundreds of thousands to even millions of years past. The paleoceanographers only have one problem, namely that if they drill a core somewhere in the deep ocean at 4 kilometers deep or so, that they have to assume that whatever they found on the ocean floor is actually representative of that location. But of course that's not true, because those foraminifera, they're at the mercy of the currents. They're planktonic, they move around. And that is what we can do with Lagrangian oceanography. We can track them back. So in this simulation, the black dot there that stays solid, that's the location of the core. And you're looking at the tip of southern Africa here, so just off Cape Town. And all of these dots are the individual virtual foraminifera that end up on that exact location. The coloring is the temperature that they've seen until that. The size is is their size, their age, essentially. And you see that sometimes they come from really cold, from the Southern Ocean. They come up north. Sometimes they come from the Agulhas current, and they're much warmer. And all of this ends up in exactly the same location in the ocean. So as a paleoceanographer, you will need to take this into account. You will need to take into account this lateral advection by the currents into everything that you find in your sediment core. Now we've done it for this location, but we could also do it for the entire globe. So this is for every location on the entire globe, the average drift distance in kilometers between the location where a foraminifera ends up on the seafloor and the location where it was born. Still, no biological oceanographer has ever called me out against a foraminifera being born, so I keep saying that. I'm not really sure if it's the right word, but what else? And you see that, well, in the subtropical gyres, where things are kind of like slowly moving around, those distances are 30 to maybe 100 kilometers or so. But as you go to the Southern Ocean, to the extensions of the Western Boundary Current, and especially to the tropics, those can be more than 1,000 kilometers, those distances. And what's more important, it's not just the distance that they've traveled. The really important metric to a paleoceanographer, of course, is whether the temperature recorded in the shell as that foraminifera moved along its pathway, how much different that is from the local temperature right above the location of the core on the ocean floor. And that's this map. The good news is, in most of the ocean, it's pretty negligible, less than half a degree or so. But as soon as you go, especially to the equator and the extensions of the Western Boundary Current, which just happen to be the locations where paleoceanographers are most interested in, unfortunately, those biases can be more than a degree, right? Which is bigger than the error of the method, the isotopic method itself. So this is something that needs to be taken into account. And this is then also the point where my talk really nicely fits into Heidi's before. Because of course, if you take a sample in the ocean, if you go out with a bucket of water or with a fancy flow cytobot, imaging flow cytobot, and you measure what is happening right there, then the water that you're measuring is not from actually, the water is from that location, but the organisms that are there have actually adapted to a location upstream. They've been washed down with the currents, right? So thinking about this intergenerationally or so on timescales, and this works especially for microbes that have a very quick turnover time, that have a dividing time of a day or so. If you assume that over 500 generations or so they can adapt to new environments, then that means that whatever you find somewhere in the ocean has actually been adapted to the last 500 days of its trajectory. You need to move this stuff back. That's exactly what we did in a paper with Martina Doblin in PNAS a few years ago. And we calculated what the difference is between the local temperature at any point and actually the temperature over a 500-day back trajectory. And that's this. In most of the ocean, and especially in the subtropical gyres, that is 5, 6, maybe 7 degrees of difference. So if you go out into the Gulf Stream and you measure something that is there, the microbes that are there, then they have adapted to temperatures that were probably 7 degrees warmer than the local temperature that you find there, just because they are at the mercy of the currents. So this is for passive organisms. This is for organisms that just float and can't do much about where they are. But I would argue that even actively swimming organisms, that organisms that can move around in an open ocean, in the pelagic environment where there's not many cues around exactly where you are, even they are to some extent at the mercy of the currents. And because they are, I think that you can actually take that and use the Lagrangian framework also to model, to simulate virtual fish. The way that it works is essentially, well, you've got your ocean, and you've got a flow on the ocean currents, and the fish or the larva or whatever you want is just moving with that current. And on top of that, it's swimming. But if the ocean would be homogeneous, if it would be the same in every direction, then the swimming will just be Brownian motion around, well, there's no preferred direction anymore if it's isotropic. The point, of course, is that the ocean is not isotropic, that there are gradients, whether it's temperature, whether it's food, whether it's oxygen, whether it's whatever you want it to be. There's one place that is a better habitat and there's another place that is a less good habitat. And we don't really know how organisms then sense this gradient, but they do. And that means that instead of just random isotropic Brownian motion, that these organisms tend to prefer going up to their better habitat. But this is a process that is really easy to tackle into mathematical equations. So we can actually simulate this in our model itself. This goes back long years, and a really nice example of this came out of the University of Miami, the group of Claire Paris, together with Erika Staterman and Judith Helgers, where they simulated how reef fish larvae move from the dry tortugas. So you're looking at the Florida Keys here, how they are swept along with the Gulf Stream past all the Florida Keys. And only very, very few, the blue dots, only very few of them actually end up on the reefs in the Keys. But if you included just a little bit of swimming, maybe 1 or 2 centimeters per second or so, not nearly as much to overcome the entire Gulf Stream, but just a little bit of swimming and directionality towards the reef, then many more reef fish larvae actually ended up on the reefs. So this was a very, very efficient way, it was shown, for connectivity between the reefs to be established, for the fish to actually end up on the reefs and to spawn new ecosystems there. Now this we can do for reef fish, larvae. We can also do it for tuna. And with a postdoc of mine, Joe Scott Phillips, we've actually made a full tuna model, a Lagrangian tuna model where they move around in the ocean. So we do that, and we did that, and we could make this tuna model because we've got a new framework, a new code that really allows rapid development of Lagrangian models, Lagrangian tools. It's called parcels. It stands for probably a really computationally efficient Lagrangian simulator. The probably I still need to stress a lot. But we're getting there. It's getting more efficient. And it's not just a model, it's more a framework. It's a set of Python classes and methods for building your own Lagrangian particle model. If you ever wanted to simulate the movement of whatever it is, of your favorite organism, of your favorite water mass, you can quite easily do that in parcels. And the way it works is that it's kernel-driven. So kernel is a particular action on a particle. A particle can be advected by the currents. It can diffuse due to random motion. And on top of that, it can do things. It can sink, particles can swim, particles can be fragmented in terms of, say, the plastic. And all of these we encode in a kernel. You can write them yourselves. They're typically a few lines of code, and then you concatenate them, and then you put that into the framework, and it just runs. And it creates these particle trajectories for you. So that's all there on oceanparcels.org. We're actively developing this. We have on GitHub 98 stars, and I thought, well, if there's 3,000 people in this room, I'm sure that we can get above 100 after lunchtime. Yeah, and this is really still active development for 5 years already. And we're doing that because we need a computationally efficient Lagrangian simulator. The title is really what it says. We need this because we as oceanographers, especially as numerical oceanographers, computational oceanographers, we're going into an age of extremely big data, the petascale age of data. Just to give you a bit of a feeling for where we're heading. Remember the very first animation I started out with, the particle moving around the lonely particle throughout all of the oceans, right, from the 1990s. That was on a 3-degree horizontal resolution. You need probably 30-day temporal resolution then. If you want a 50-year simulation of that and you want to store that on a computer drive, it's a few gigabytes of data or so. Then as you up the horizontal resolution, at some point you also need to up the temporal resolution because the eddies are going to be more energetic and more varied, so you need to save the data more often. We're now at the stage where we have regular 1/10 of a degree or so simulations, 5-day resolution. Typical simulations for 50 years of data is a few tens of terabytes. But people are already planning simulations at a 50th and even at a 100th degree global simulation. And if we still want 50 years of data, and that's important because only on 50 years can we really understand connectivity on a global scale. If we still want that, then we get to petabyte simulation. So very soon, we as oceanographic community, we will need to start thinking about how to deal with the petabyte age. How do we make sure our tools and infrastructure are ready for this? Well, fortunately, there's a group of very, very smart people thinking and worrying about this for you, so that you don't have to do it, or with you if you want to think with this. It's called the PanGeo project. It's a community that really is very active in making sure that we are ready for this petascale age, that we have the tools to deal with this. So that's the how do we do it in terms of just the codes and the computation. But I think there's another thing. How do we make sure that whatever we're going to do on these models, that it is shareable and that it is reproducible? And that, I think, is a much harder problem. How do we make sure that whatever we do is reproducible, is verifiable, that we are accountable, essentially, for what we're doing? And for that, I think we need a radical rethink. I think we need to go towards a radically open science. So many of you, hopefully, already practice open science. And open science, for most of us, starts with open science of analysis. That is sharing code, that is sharing data, that's sharing notebooks, that's maybe even pre-registration. Probably some of you are already also doing open writing on platforms like Overleaf, for example, and creating notebooks to open up the way you do your analysis. Then many of you, of course, have heard of open access. So open publication, but it's not only open access, it's also open peer review and sharing preprints to make sure that we are open in that. Then there's outreach. So the communication via social media that we're doing, sharing the posters, this conference is also supporting that, where we really share whatever we're doing. And there's open assessment about alternative evaluations, things like altmetric and these kind of things. So that hopefully most of you are doing already. But I think there's also on the other side, there's something that we can do more. There's the open search, where we share and we comment on each other's papers openly, where we share each other's library. And then there's the last step, and this is the most scary one. This is the one that I am not even really yet ready to do. I think we ought to do it, but it is scary to actually do it. And that's the open proposals. You know, in an ideal world, if I write a proposal and I submit it and it doesn't get funded because apparently I am not the right person to do it, then shouldn't I just make that proposal open and hope that somebody else actually takes it up and does it? Shouldn't there be somebody, probably there is somebody better out there. One of you can do the things better than I can, right? So why should a proposal be mine? Why should I have so much ownership and feel so much ownership of my own proposals? Right? Aren't we all here in the same game? Aren't we all doing science because we want to advance science? Isn't science far too egocentric? Right? So I would love the day where it is completely normal to share our proposals, to work together on our proposals, to be completely open by that, and not having to worry about our own careers. So that is a radically open science, where we plan our research through co-creation, where we conduct our research through transparency and inclusivity, where we communicate with peers and the public, and importantly, where we get recognized and rewarded in an unbiased way. And that last thing, that's the trickiest, because that's not in our hands. That's in the hands of our university administrators, that's in the hand of our funders. They need to make a change into how we get recognized and rewarded for what we do as academics. And I'm really, really happy that at least in the Netherlands, there's now a very strong push for changing this. It's called Room for Everyone's Talents, and it is really about how to keep sane and healthy in academia. Because there's far too much over effort. There's far too much stress among each other. There's far too much burnout in our community. We need to do something about that. And the way that in the Netherlands we're thinking about this is that we have 4 pillars: education, research, impact, and leadership. And the idea is you don't have to be excellent in all. Nobody's excellent in everything. We are not training sheep with 5 legs. So everyone can pick some things that they're better in and some things that they're worse in or less good in, right? And we need to tell that story together. Then, on top of that, we need to think more in teams. So we need to really achieve a balance between the individual and the academic. Assessment shouldn't be about individual persons. It should be about the teams that they're part of, like any normal company would do. It shouldn't be about numbers so much. We are far, far, far too focused on things like impact factors, H-scores, citation metrics, all the things that we can measure. If we can measure it, we think that it is appropriate and that it's correct. But there are enormous biases in these measurements. So we need to go to a system where it's much more about the narrative of our research, about why we do the research that we do and why we think it is important that we're doing. That's the way that we should start thinking about the rewarding and the recognition. And finally, we really need to, for everyone and on every single level, think about stimulating academic leadership. This is not something for the big professors, for the dinosaurs. Everyone should take leadership and should stand up. So, after that little rant, and I hope you're okay with that, let's go back to physical oceanography. The ocean is no bathtub. It's in constant motion and full of small-scale eddies, which have a crucial role in the transport of heat, nutrients, plastic, and plankton. I've shown you that most of the plastic in the ocean is missing, and that mapping this plastic is an exciting challenge for oceanographers because plastic is a unique tracer. And I've talked about Lagrangian ocean analysis in general as a way to really understand ocean connectivity and conditional pathways and how everything in the ocean is connected, because in the end we only have one ocean. But to cope with all of this, we need to reward radically open science. Thank you very much, and thank you to the team.
Thanks, Eric. Okay, we have time for a couple of questions before y'all head out to lunch. So again, we got our 1, 2s, and 3s up here. And I'd particularly like to encourage students, early career, anybody? Yeah, please.
Hi there. Thanks for an amazing talk, interdisciplinary. And while it's very clear that we need to learn more about the transport of plastic in the ocean, we also know a lot about the inflow of plastic. And so in the meantime, we need to work on shutting off the tap. So as somebody who has clearly thought a lot about this, if you could choose one sort of change or one legislation or one shift in society that would have the greatest reduction in our outflow of plastic to the ocean, what would that be and why?
Yeah, good question. so now I'm really stepping beyond my remit as a physical oceanographer, of course, but I talk to many plastic people a lot. I think in the end, so we have plastic in the ocean because our waste management system is broken. It's as clear as that, right? There's bad waste management, and therefore plastic ends up. Nobody goes out there and tries to put plastic in the ocean just for the fun of it. So why is our waste management system broken? Well, there's many, many reasons of that. But if you think about what is the easiest way to fix it, I think start with product design. I think that, I hope there are not too many people who are product designers in this audience, but a lot of the blame is on their shoulders, right? Why does every single soft drink bottle need to be different? Why does it have to be a different color? Why does it have to be non-recyclable? Why does some plastic need to be black? Most of the recycling that we do is done on infrared, on spectral information. Black plastic doesn't reflect, so we can't actually recycle it through automated processes. And still, fancy things are packaged in black plastic. So that would be the place to start, I think, legislation-wise. Easier plastic.
Okay, great. We're going to go to Mike 2 for the next question, please.
All right. Well, thank you for that talk, and I really appreciated your focus on open science at the end. I have a few technical questions about your Lagrangian particle tracking. So, first of all, how do you interpolate velocities within the model, and then how do you validate the model tracks if you...
Yeah, good questions both. So the interpolation, we can choose. We have different interpolation schemes. My postdoc, Philippe de Landmeter, wrote a very complicated paper in GMD about exactly how we do the interpolation. It depends a bit also on the grid, whether it's A grid, B grid, C grid. And even here, I've already had many talks with many people who say that our interpolations are still wrong. So, open question, I don't know. In terms of the validation, well, the gold standard is still buoy trajectories for the surface at least, drifting buoys, Stokes drifters that really sit on the surface of the ocean. We're doing more and more of these. In 3D, that is difficult. It's difficult to really validate these. But in the end, I think the bigger errors from the ocean hydrodynamic model than from the actual particle trajectories. I mean, in the end, it's just an RK4 integration, right? It's interpolation and multiplication. It's not much more than that.
All right, well, thank you.
Yeah.
Okay,
Hi, I'm Peter Kalmas from JPL. Thanks for the talk. And thanks for the rant on the radically open science. I think it's good to step outside of our discipline sometimes and think about how we organize. So this is kind of a question or comment on that, which is I think probably the majority of us in this room would agree that the planet is somewhat in peril right now. And how can we also change assessment and the reward structure to encourage more scientists to kind of raise that alarm and get the public to kind of support policies that we all know that we need to start slowing this down and then fixing it?
Well, that is exactly my point. Thank you for raising this so well. I think that in our old reward and recognition structure, where it's just about impact factors, just about H-scores, it doesn't actually recognize if we do something meaningful. If we put our efforts into actually changing this Earth and this planet that we live on. So I think that exactly for that reason, we need to just change the system and reward impact more. Ask people at their annual performance review not how many papers they've published, but what their papers have meant to policymakers, to change makers, to stakeholders. Those are the important questions to ask.
Okay, last question.
Erik, a great talk as always. So I thought one of the goalposts or something like we could do together in the next 10 years that also this radical open science could enable is, I think you had the beginnings of what I'm calling a digital twin ocean.
Yeah.
It's sort of the digital analog of the real ocean. And I think it has all the elements that you showed us. Because if you can track what moves through that digital ocean, you can learn something about it. If you can image what's there with flow cytometry and other ways, we can explore the ocean. Yeah. but are we not really working to that one big goal to try to take our real environment into the digital space so we can dial it back and forth in time, we can zoom in space, and we can also dial in issues like where's the plastic, where the fish, where the phytoplankton, where the risk map. Isn't that something that inspires you?
That's exactly the point, this digital ocean, and we've talked about this before, Martin. There was a fantastic tutorial yesterday that I attended about a package called Spy Ocean that comes out of the Pangeo stack that does exactly that. It is a ocean-going software without actually having to go into the ocean. You can put your moorings into a virtual ocean. And this digital twin that we can manipulate, that we can test hypotheses in, that we can investigate further, is fantastic. That is what we need to build. And the Pangeo people are certainly building that, but they need help. They need more people, and especially they need more money.
Always. Great. Thank you. Please join me in thanking our speakers this morning. All right, I hope you guys have a good afternoon, and I hope to see you at the jam session tonight and the fun run walk tomorrow morning. Extra bragging rights to those of you who do both. Have a good meeting.